Today we'll discuss about fertilization. Before studying about fertilization, let us discuss about coitus. Coitus is also named as sexual intercourse. Sexual intercourse. Even it is also named as copulation. Coitus or sexual intercourse is necessary in humans. That is mainly because of there is a internal fertilization. Internal fertilization. That is, the fusion of male and female gametes takes place in the genital system of females. That is internal fertilization. Because of internal fertilization, it is necessary as far as coitus or sexual intercourse. Coitus is a process by which a sperm from the male is introduced in the genital system of female with the help of penis and is said to be insemination. Insemination means introduction of semen into the genital system of female during the process named coitus with the help of penis is named as insemination. At the time of insemination, the male genital organ, penis, has to get erected. Penis has to get erect. What makes the erectile nature of penis? Penis receives blood through the arteriole. Blood comes to penis through arteriole and goes back through venule. At the time of coitus, sexual intercourse, arteriole undergo dilation. Dilation in the sense, those arteriole blood vessels get enlarged so that more blood comes towards penis. And at the same time, venules get constricted. It means they are compressed so that minimum blood has to go back. Due to dilation of arteriole, maximum blood comes towards penis. Due to constriction, minimum blood has to go back through the venule so that high blood pressure is created in penis. Once the BP is increased, the penis becomes erect. Once it becomes erect, it is very easy to introduce the penis into the vagina at the time of sexual intercourse. And the erectile nature based on dilation of arteriole and constriction of venules. Already you know that blood comes to penis through arteriole and goes back through venules. At the time of sexual intercourse, arterioles get dilated, enlarged, so that more blood flows towards penis, and venules get constricted so that minimum blood has to go back. so that high blood pressure is created in penis, and that leads to erectile nature of penis, so that it helps for coitus or sexual intercourse. It's about the coitus or sexual intercourse. By this, semen is introduced into the uterus, and during that sexual intercourse, they will reach, sexual partners will reach a climax. That climax stage is named as orgasm. Orgasm. They reach the climax, and that is the time the semen is ejaculated. Already you know that. Once the semen is ejaculated, that semen is around 3 ml. In this 3 ml of semen, there are around 200 to 300 million sperms are there. In the 3 ml, 200 to 300 million sperms are there, in which 60% of them are with definite structure, size and a shape, and 40% of them are vigorous motility. They can climb, they can move vigorously, 40% of those sperms. And now it's time to discuss about a process of fertilization. A process of fertilization. Let us see about fertilization. Fertilization is a process where haploid sperm fuses with haploid egg or ovum to form diploid zygote. In case of humans, it is taking place in the fallopian tube, internal fertilization. Here I have drawn the structure of female reproductive system. Already we have studied about the structure. It's a ovary, a female gonad, and this is fallopian tube or oviduct, and it is having 3 distinct parts. Those are infundibulum, by which it attaches to the ovary, and ampulla, and the part which is connected to uterus is isthmus. Infundibulum attaches to the ovary with the help of finger-like projections named fimbriae. And this connection is temporary. Once the pregnancy is achieved, fimbriae are separated from ovary, as it is temporary connection. And now this is uterus or womb, and its opening is named as cervix. Cervix leads into a female genital organ, vagina. During sexual intercourse or coitus, the semen is introduced into the uterus. There would around 3 ml of semen comes to this uterus. Already I have mentioned Around 200 to 300 million sperms are present in it, in which 40% of them are vigorous in motility, so that thousands of them move towards this fallopian tube, thousands of them move towards this fallopian tube. And by the time, during middle of menstrual cycle, that is on 14th day, egg is released. That egg is released from the Graafian follicle. Once that egg is released, that egg through the infundibulum, it moves and comes to the place, that is the place where fertilization takes place. That is the place which is there in between, in a junction between isthmus and ampulla. That is said to be ampullary isthmic junction. Ampullary isthmic junction. They may ask one last question. Which part of fallopian tube fertilization takes place? That is the junction between isthmus and ampulla. That is ampullary isthmic junction, where slightly that fallopian tube is enlarged, and that is the place where fertilization is possible. Sometimes, by the time egg is here, but sperms will come up to this. They face each other. No chance for fertilization. Sometimes that egg might have come in isthmic region. Sperms will come. They face each other. No chance for fertilization. Sometimes the egg comes to uterus. Sperms will come in, face each other, but no chance for fertilization. Only at one place fertilization is possible, that is ampullary isthmic junction. Ampullary isthmic junction. And thousands of sperms, they move. In both the fallopian tubes, and one is always ahead. That touches the outer membrane of that egg. This is egg or ovum and is a sperm. Thousands of them are moving towards. And sperm, one among them is always ahead, that touches the outer membrane of ovum. Already we have studied the structure of ovum. It has its plasma membrane. Next to that is vitelline membrane and the spacious zona pellucida, and outermost corona radiata. This is corona radiata. This is ovum and outermost corona radiata and a spacious zona pellucida, and one more additional membrane, vitelline membrane, and innermost is a plasma membrane. In that antillary isthmic junction, many sperms have come and touches the outer membrane. Already we know that head of sperm is having a acrosome. In that acrosome, a lytic enzyme like hyaluronidase is there. A enzyme hyaluronidase is there in that acrosome. Once it touches, acrosome burst and hyaluronidase spread over the corona radiata. And these corona radiata cells are packed with a chemical substance named hyaluronic acid. When head touches, lytic enzyme spread over the corona radiata, and the lytic enzyme is hyaluronidase in the acrosome. These corona radiata cells are packed with chemical substance. That chemical substance is named as hyaluronic acid. By the action of hyaluronidase, this hyaluronic acid gets dissolved. It gets dissolved. Due to this, some of corona radiata cells are replaced. They are detached, removed out, and this sperm can penetrate it and enter into ooplasm of ovum. That enters into ooplasm of ovum. And once that head reaches the ooplasm of ovum, some changes are taking place inside. This is plasma membrane with ovum nucleus. Next to that is vitelline membrane. Here one change has taken place. That change is the space between plasma membrane and vitelline membrane. This is said to be perivitelline space. That perivitelline space is increased. That perivitelline space is increased. And the head, it has entered into the ooplasm. Around the head, plasma membrane makes a cone-shaped structure. Only around the head, plasma membrane makes a cone-shaped structure that is said to be fertilization cone. Fertilization cone. Then this space is also increased, and here zona pellucida is there, and outer corona radiata cells are there, and the place where sperm entered, corona radiata cells are not there, and some other changes are taking place now. That is, this plasma membrane becomes permeable. Plasma membrane becomes permeable, so that pigments present in ooplasm, they come out and deposit on vitelline membrane. and vitelline membrane becomes thick. Vitelline membrane becomes thick. And now this vitelline membrane, it is thick because of the pigments present in ooplasm are crossed through the plasma membrane, deposited on that vitelline membrane, and now this vitelline membrane becomes fertilization membrane. Fertilization membrane. Once vitelline membrane becomes fertilization membrane, it is a barrier for other sperms to enter in. Other sperms, they are unable to enter in because this fertilization membrane is very thick. So that here thousands of sperms have entered. One is always ahead. Only that sperm entered in, and the rest of the sperms, they too come. Even they can come up to this. Maximum they can go up to vitelline membrane, fertilization membrane. They are unable to enter into the ooplasm. This fertilization membrane mainly helps for monospermy. What is monospermy? Entry of single sperm into the ooplasm. That is said to be monospermy. And it prevents polyspermy. It prevents polyspermy. It means entry of many sperms into the ooplasm. That is said to be polyspermy. What is monospermy? Entry of single sperm into the ooplasm. That is very possible because of fertilization membrane. Polyspermy means entry of many sperms into the ooplasm. That is polyspermy. But due to fertilization membrane, that polyspermy is prevented in case of humans. But we do see polyspermy in case of birds. In birds, many sperms can enter into ooplasm. They may ask a neat question: polyspermy is possible in which of the following animals? Birds. But in case of humans, monospermy is possible. That is mainly because of fertilization membrane. And here they can ask some neat questions. Fertilization cone is formed by which of the following membranes? Plasma membrane, vitelline membrane, zona pellucida, corona radiata. Answer is that is formed by plasma membrane. Which of the following membrane becomes fertilization membrane? Answer is vitelline membrane. How? When head of sperm enters into ooplasm, the space between plasma membrane and vitelline membrane is increased. Then cone is formed around the head. Pigments present in ooplasm are passed through plasma membrane and deposits on vitelline membrane, and the vitelline membrane becomes thick. Now that vitelline membrane is said to be fertilization membrane, and it becomes a barrier for other sperms to enter in. Sperms can come only up to fertilization membrane. Only one sperm can enter in. Due to fertilization membrane, monospermy is possible. It prevents polyspermy. And now head entered inside. In next step, this is female pronucleus present in ooplasm, and a nucleus brought from sperm is male pronucleus. Male pronucleus. A nucleus of the ovum is female pronucleus, that is having 23 chromosomes, one set of chromosomes, one set of maternal chromosomes, and a nucleus introduced by the sperm is male pronucleus. It is also having 23 chromosomes, but those are 23 chromosomes of paternal set. And what happens is their nuclear membrane get dissolved, and those chromosomes, maternal chromosomes and paternal chromosomes, they get paired, and a common nuclear membrane is formed. Once paternal set of chromosome and maternal set of chromosome get paired and a common nuclear membrane is formed, now this nucleus becomes diploid and this ovum becomes zygote. Hence, fertilization took place. As soon as female pronucleus and male pronucleus are there in ooplasm, they come close to each other, their nuclear membranes get dissolved, and maternal set of chromosome and paternal set of chromosome get paired, 23 maternal chromosomes, 23 paternal chromosomes, they get paired, a common nuclear membrane is formed. Now this nucleus is diploid and the ovum itself becomes zygote. Ovum itself becomes zygote. Hence the fertilization. Here, one more thing we should notice. While telling about a process of oogenesis, I have mentioned process of oogenesis is very complicated. It starts from the embryonic state of a female up to a time of fertilization. When the sperm touches the outer membrane, at that time this is not the ovum. Actually that is secondary oocyte. Now that secondary oocyte undergoes meiosis second, leading to the formation of ooted. That ooted becomes ovum. And another one is second polar body. That second polar body either may get degenerated or remains as a non-functional one. Remember, a process of oogenesis completes at the time of fertilization. When a sperm touches the outer membrane, that the secondary oocyte undergoes meiosis second, forming larger ooted or ovum and a smaller second polar body. But in general we have taken it as ovum because already meiosis second took place. Okay, it's about the formation of zygote. Once the zygote is formed, now that zygote starts undergoing the first division, that is cleavage. A repeated mitotic division of zygote is named as cleavage. Once the cleavage takes place, a single-celled zygote becomes two-celled, four-celled, 8-celled, 16-celled. By the time that zygote comes to uterus, it would be 8 or 16 celled. 8 or 16 celled. Let us see how these developmental stages come across when that zygote comes to uterus. And now, once the fertilization takes place and a zygote is formed, this is a single-celled zygote. Now this zygote undergoes cleavage. And the first cleavage starts from animal pole to vegetal pole. It becomes two-celled. Is a first cleavage that is said to be meridional division. It becomes two-celled. And next cleavage is already first meridional, second one is also meridional, but at right angles to first one is also meridional and is four-celled stage. First two divisions are meridional. It means you keep apple. If you cut from here, two pieces are formed, equal pieces. That is meridional division, and another one at right angles to first, meridional, four cells are formed. That is the stage. And now third cleavage is already two meridional divisions took place, and third cleavage is latitudinal. Latitudinal in the sense transverse, latitudinal division. It becomes eight-celled. First one meridional, right angles to this is second meridional, and now transverse section means latitudinal. It becomes eight-celled. Here, latitudinal division is not at the middle. It is towards animal pole. So that four smaller blastomeres are formed. Those are named as micromeres. And four larger blastomeres are formed. Those are named as macromeres. In 8 cells, all the 8 cells are blastomeres. But towards animal pole, they are smaller in size. Those are named as micromeres. Towards vegetal pole, those 4 are larger in size, named macromeres. And now, this 8-celled stage becomes 16-celled. Micromeres towards animal pole and macromeres towards vegetal pole. And this ball of cells is named as morula. This ball of cells is named as morula. And in this morula, all the cells are pushed towards periphery. And forming the layer named trophoblast, and group of inner mass cells. Inner cell mass, and this inner cell mass becomes the embryo, and a trophoblast is meant for the attachment of this embryo to the endometrium of uterus. Is meant for the attachment of endometrium of uterus, trophoblast. And now this stage is named as blastocyst. This stage is named as blastocyst. See here, zygote undergoes cleavage. Cleavage is nothing but repeated mitotic division. By first cleavage, a meridional division, zygote becomes a 2-celled, and by second meridional division, which is at right angles to first, it becomes 4-celled, and a third division is transverse latitudinal, so that 8-celled stage, but that latitudinal stage division is not at the middle, it is towards animal pole, so that 4 smaller micromeres and 4 larger macromeres, so that blastomeres are differentiated, and then it leads to 16-celled, and micromeres are there towards animal pole and macromeres are there towards vegetal pole, and that ball of cells formed is named as morula stage. And now in morula some changes taking place. Cells are migrated towards periphery, forming the layer named trophoblast, but at the center a group of cells are remained, that is inner cell mass, that becomes the embryo, and this stage is named as blastocyst. And this stage, blastocyst, attaches to the endometrium of uterus. This attaches to the endometrium of uterus. It is embedded inside. The blastocyst embedded in the endometrium of uterus, which is named as implantation. Is said to be implantation. This is morula, blastocyst, and it attaches to the endometrium, and embedded in endometrium itself, this process is named as implantation. Once blastocyst attaches, implantation takes place, it means the pregnancy is achieved. At this time, the pregnancy is achieved. Once the blastocyst attaches to the endometrium, implantation takes place. Once implantation takes place, the pregnancy is achieved. And here in endometrium, that blastocyst outer layer forms a finger-like projections named chorionic villi, and even uterine tissue, they are interdigitated to form a placenta. They form placenta. That we will see later on. The blastocyst embryo which has attached to the endometrium, is it a male embryo or a female embryo? That is decided not by the ovum but by the sperm. For that, we must know the nature of genotypes of male and female. Male is having genotype 44 autosomes and 2 sex chromosomes, X and Y. And female is having genotype 44 autosome and 2 sex chromosomes, those are X and X. Genotype of male is 44 autosome, X and Y sex chromosomes, and a female is 44 autosome and X and X sex chromosome. From this, two types of gametes are formed. In 50% of sperms, 22 autosome and X is there. In the other 50%, 22 autosome plus Y is there. There are two types of gametes are formed from male. 22 autosome plus X, 22 autosome plus Y. And here it is a female. From this, only one type of egg or ovum are produced. All of them are having 22 autosome and X, because both the sex chromosomes are X. As two types of sperms are produced by male are said to be heterogenetic. But females produce only one type of gametes, hence it is homogenetic. They are homogenetic. See here how the sex of embryo is determined. This sperm, in case fuses with the egg, now it becomes 22, 44 autosomes, X and X. This is a female embryo. Instead of this sperm, a sperm having Y fuses with egg or ovum, 44 autosome plus X and Y. This is male embryo. A sperm having X fuses with the egg, embryo becomes female. A sperm having Y fuses with the egg, the embryo becomes male. So that whether the embryo is male or female is decided by the sperm but not by the egg. The gender of a child is decided by the male, not by the female, because a type of sperm which fuses with the egg decides it. A sperm having X fuses, that becomes female. A sperm with Y fuses with the egg, that decides the male embryo as well. And now, once the pregnancy is achieved, during the pregnancy period or gestation period of 9 months 10 days, what are the changes we do find in them, and also how this placenta is formed and what are its function, that also we have to see now.